So we are among friends, and what I'm going to talk to be, it's a little bit different from the title as usual, because I think that among the people that are in the Consilium consortium, let's put it this way, precision oncology is really a hot and controversial topic, and with some that are advocates and some that are critics and says that it's clinically relevant. I think that we all agree that the one drug, one care fits all model needs fixing. And I posit that precision oncology is large enough to cover both research and care. And I will try to support this this thought with the past and current efforts from my group in colorectal cancer, focusing on the translational platforms that I designed in the last 10 years on liquid biopsy and on the fact that primum non nocere. Precision oncology is already gone mainstream for a while, and it's recognized by the FDA at various levels of approval, both in the treatment and in the standard of care resistance biomarkers. And it's also financial toxic with a spatial heterogeneity due to the fact that everything in the USA costs twice as much as it costs in Italy. And these are all data on the left and new data on the right. And in any case, even for the European, the cost of these type of treatments is higher or equal to the salary, so the average salary. So we're talking about something that has a big impact in also socially. And this, so the question that we all have is it worth? Is all this cost worth? And this is the data from a very controversial JAMA article that came out a couple of years ago, showing that really less than 10% of the patients were eligible for genomically targeted treatment, and even less than 5% were getting benefit from targeted treatment. Now, this is really a little bit a negative side, because there are a variety of caveats that one can do, but still, even if we say that this is too negative and we do double that, it might be still a problem, especially looking at the matching cost. More recent data from 3 important trials that came out a year and a half ago, showed that perhaps there is more meat in precision oncology, because here, this is second generation precision oncologists, i.e. targeted treatment that use either drug combination and not monotherapy, targeted therapy using picked up by ctDNA sequencing, or combination with immunotherapy. And since these are all early clinical trials, i.e. patients that were mostly overtreated and resistant, response rate going from 10 to 23% to 36% might have an interesting new perspective. On the other hand, the problem is the feasibility of this type of approaches, which, as you can see here, is really very, very small. So I don't want to discuss all the attrition of precision oncology, with perhaps the exception of these 3 in red, but there are many of them, and I've just listed some of them. I'm interested more in the how would I say, the one that have either a conceptual flair or a more anthropological one. But in order to try to discuss these things, I wanted to start at the beginning. I started where I started 10 years ago, when the relationship between basic science and clinician was more or less as is in this picture that was published in Nature in 2008. So scientists on one side, clinician on the other one, and the patient dead in the middle. To overcome this, we created in 2011 an ARC colorectal cancer consortium. ARC is the equivalent of CRC UK, of Cancer Research UK charities, the most important charity in Italy. Without them, we wouldn't have gone anywhere in Italy in cancer research. And to infrastructure this consortium, I created and designed and maintained 3 subsequent translational platforms. ACA, Master Observational Trials, Razel Curzo calls them the MOT, and I like it because it's a short name. So my 3 MOT in sequence were profiling, which we did to establish models that could phenocopy human cancer. And funnel subsequently started in 2050, lasted only 3 years, that allowed us to link the clinics to the labs for doing NGS and NGS screening. And eventually, the one that is going on now, Alpha Omega, which is something that I designed to tackle complexity. And so the RCC team is made by a research network, which is headed by Alberto Bardelli, who is also the PI of the entire consortium, a clinical network, which is held by Salvatore Siena. We're now 12, we grew during the pandemic. So we used to be 10 and we're now 12. I didn't have the time to make the slides. And then in the middle, it's me, the translational hub, and I'm the head and the coordinator of the translational hub. So in other words, what my lab does is it's the chocolate between the 2 noisette biscuit that keeps together the labs on one side and the clinic on the other. And the goals of the consortium were quite simple. We wanted to do research on CRC, on colorectal specific unmet clinical need, and we wanted to bring the results of this research back to the CRC patient as soon as possible. And to do this simple thing, we did a very complicated things that I will not go into it, but certainly it was kind of a virtuous circle, as I like to call it a little bit bombastically, in which we started, getting the sample from the patient and then transform those in experimental models, then do trials in the models, and then have rationales that we validated in retrospectively in the clinic, and then we do proof of concept trials, and then from there we derived sample and data and started all over again. And in the middle, there are the platforms. Okay, so the first one was to establish models that phenocopied human cancer. And the idea was to create avatars of patients, and many of them, because we wanted to take care and look at heterogeneity. And so we did that by, this is a very old slide. We have now gone above 1,200 avatars of patients in PDX. They're not all online, obviously. And we also did, we're now doing PDOs and cell lines, human cell lines. And all these are quality controlled. There's a lot of sequencing going on. There is a core of these in which we have 250 approximately, in which they have whole exome, and in a subset of that, also whole genome, and then epigenetics, and so forth and so on. Livio Trusolino and Andrea Bertocchi are responsible in Turin for this. And the first results of this tremendous effort that took us 5 years was to create the first thing that we did was we tried to prove that we were phenocopying what was happening in the patient. We did a trial with cetuximab. So this is the waterfall plot of a trial in mice. Each line is an avatar in which, as you can see, we really looked exactly, phenocopied what happens in man with Xeno, with anti-EGFR treatment. Very few patients respond, around 20 to 30%. The majority is either stable disease or in progression. And within the progression cohort, we picked up by screening molecularly this patient, 5 patient avatars that were HER2 amplified. And on the basis of that, we did a trial in these patients, in avatars, not in the patient, in the mice, and figured out that we needed to do a double blockade of the HER2 gene, with lapatinib and trastuzumab. We did several trials. And then we went to clinic because the results were so good. Before going to clinic, we developed our own diagnostic algorithm, and we screened 1,200, more than 1,200 patients to get 69 positive, 5.4%, which is smack on the middle of what is the HER2 positivity confirmed later on by other people. And of these 44, we enrolled in the two trials. This is also older. And this is, in a way, the proof that precision oncology stands on exception. And if we don't find a way of making things simpler, it's not going to go anywhere. The trials was extremely positive. We have an objective response rate of 30% and a disease control rate of 70. Remember, these are, median 5 pre-treatment. We had 2 CRs, and one she's still alive 7 years after the treatment. This is really something that you achieve only if you have a tumor that is truly addicted to HER2 with more than 10 copy numbers. And just to put the things in perspective, this 30% response rate with a simple treatment that costs relatively low, it's not very different from the immunotherapy with monoimmunotherapy with pembrolizumab, which is hailed as the best things after sliced bread in colorectal cancer MSI patient. And in 2009, the NCC included the HER2 target therapy in the guidelines for standard of care. So from there, we went to, in the meanwhile, to the second platform. We enlarged and embellished the platform with the idea to link the clinic to the labs and vice versa. And we created Funnel. Funnel was only metastatic patient, but was already quite sophisticated. What we were doing, we were collecting patients at first diagnosis of metastatic diseases, whether they were d'emblée or because they were relapsing from adjuvant, and collected a lot of stuff that you can see here. We NGS'd with a little panel of our device, the paraffin block, and then started treating patient normally, and at each main clinical event, either the response or the progression, we repeated this collection, because the point here was to try to figure out what was the evolution of this patient. We also had rapid post-mortem protocol at the end, but I will not discuss, which is very useful, but I will not discuss about that. Okay, with this platform, we did 12 genomic-driven trials that were embedded in. Most of them were also commercially, because we couldn't do all the academic trial, albeit we did some. But the one I want to talk to you is Kronos, which was in the multiple wild type patient population. So those didn't have all this kind of things. Okay, as you know, because you're not oncology, let's put it this way. Metastatic disease, colorectal cancer, the mainstay of treatment in 50% of the population, which is not KRAS mutated, so it's wild type for KRAS and BRAF and all the others that I've shown you before. Anti-EGFR therapy is the standard of care on top of chemotherapy, the backbone of chemotherapy. But unfortunately, practically all the patients end up being recurring in this disease. It's an indolent disease, but they will recur. And those that recur mainly develop resistance to EGFR, which is RAS mediated or mediated by the ectodomain mutation of the EGFR receptor. And we knew that because we started following, thanks to Funnel, all the patient with liquid biopsy. And we could, as you say here is the patient that it's treated, responds, and then progress. And you can see here, this is the, I would say, the APC trunk mutation. But what is interesting is that while the patient responds, the trunk mutation goes down. And after a while, however, a KRAS, and particularly the Q61H mutation starts to come up. And then when you stop, what happens? We started asking this question. What happens when you take away the Darwinian pressure of the treatment on the cetuximab-sensible population. And what happened is that the KRAS clone goes back down. And if you stop the treatment with if you stop the pressure, and goes back up, or a new one reappear, if clinically you just decide to treat the patient, which is done, because this this idea of doing the rechallenge is something that has been empirically been done for a long time, and there has been patients were treated like that just because the drug was available, let's put it this way. But then there were a couple of trials, one Italian, that showed that in reality you could achieve a 20% response rate in this patient. Mind you, these are third, fourth line patients, therefore the response rate to chemotherapy or any other thing is below 5%. We started to ask the question, can we do it? But doing not empirically, but driven by by science, by the ctDNA. Okay, I guess if there aren't, everybody is familiar with the concept of circulating tumor-free DNA that comes from the tumors. I hope so. If not, you will ask me the question. So we designed Kronos, in which we did the rechallenge only in patients that had responded to first-line EGFR therapy with chemotherapy, had progressed, then had the intervening therapies. They were screened by liquid biopsy, and if they were RAS-BRAF and EGFR-SCD wild type, they were treated with panitumumab. We just closed this, and I'm going to show you things that I shouldn't be showing you, because they're going to. We are going to the ASCO oral GI presentation. And so we screened 52 patients. And the first things that were eligible for the clinical, let's put it, rechallenge. So these are patients that in normal circumstances would have been, all 52 of them, be treated with the rechallenge. And out of these, interestingly, 31%, so 1/3 of these patients that would have received anti-EGFR therapy in any case, they were all had still some RAS or EGFR mutation, as you can see here. So we can say here that, you know, we have spared 1/3 of the patient a treatment that for certain was not active and possibly toxic. And here's the response, which is much higher than the other one, but this is kind of, you know, I shouldn't even dare to say this in front of Ian. This is an historical control. So, and as you can see here, the duration of the response is not optimal, but this is something that can be ameliorated in the future. So, two take-home message, primum non nocere. In this case, liquid biopsy helped eliminating patients that shouldn't have been treated and that would have been treated otherwise without the liquid biopsy. And the second one is that it is an interesting response rate that we might be able to ameliorate the response. Keep in mind that these patients have no other alternatives, and the most used drug in this population is regorafenib, which in my opinion is one of the worst drugs ever commercialized because it's supremely toxic and supremely non-efficacious. I still have to figure out how they did they got the NDA. Okay, so move to the third point, embrace complexity. That is, Alpha Omega is the platform that we have now, which is actually two platforms. The one is Alpha Omega Retro, which is looking backward, trying to do retrospective archival collections that could allow us to do large numbers for marker validation studies. We would not go into the clinic without first validating the markers, as we did for the HER2. And then Alpha Omega Prospective, which is actually a movie that follows all the patients. This is on real world patient. Any patient that walks in the hospital that participate can enter here and enters here, and it's followed either to death or to 5 years NED, because they can enter also in early disease. This is a big change compared to funnel. And we do and collect a variety of things. Again, for the sake of time, I will, if you're interested, we can have separate conversation. We're collecting practically everything, and we're keeping collecting this. And we also have one of the problems that we had with Funnel, and that talking, with Caroline Dive and other people that have platform that comes out, is that this platform, if you ask everything to every patient in every hospital, they become so rigid that at the end nobody's participating. So Paolo, one of my collaborators, had this tremendous idea of doing a tier informed consent situation in which there are 3 things that are asked to everybody, which are paraffin blocked, 1 liquid biopsy and one imaging, and everything else. This is the first informed consent, and then there are several other informed consent that you can add on and modify, and certain centers can do only 1 or 2 for each of the patient. You can do a mix and match. So this allows a lot of flexibility and allows us to really do things also at a relatively small cost. Okay, so embedded in Alpha Omega, there are 4 trials, 2 ongoing and 2 which we are at the late stage of negotiation. And I will talk about only one of these, the Pegasus trial, which is a personalized adjuvant therapy, personalized on the basis of interventional liquid biopsy. So we are deciding how to modulate treatment on the basis of what we see in the liquid biopsy done 3 weeks or 4 weeks, 3 to 4 weeks after surgery. Of course, if there is still ctDNA in the blood of this patient, given that the DNA in the blood has a 2-hour half-life, that means that unseen by radiology and by CT scan or MRI, that patient has micrometastatic disease somewhere in the body. You don't know where, but it's there. And this patient have a practically 100% chance of relapsing if untreated within 2 years. Okay, so again here I come from the Marioneghi, so primum non nocere is something that was kind of stamped in my brain when I started working there. You do not have to harm. Do not harm. Do not harm with iatrogenic disease. Now, this is particularly cogent for colon cancer because we have 1 million cases a year, 50% of them will be fatal, and therefore the curative treatment of stage 3 colon goes through the combination of surgeries plus adjuvant chemotherapy. And should I not know, one of my first trial in colon cancer, a year ago, and I don't want to say it because you wouldn't understand how old I am, was to prove that you need chemotherapy, adjuvant chemotherapy. At the time it was FUFA, 5-FU and folinic acid. You can tell how long ago was that. But unfortunately, in reality, 50%, fortunately, in reality, 50% of these stage 2 high risk and stage 3 patients are cured by surgery alone, However, we don't know which is which at the time of surgery. So we have to treat everybody with an oxaliplatin-based regimen that impairs the patient quality of life because of neurotoxicity, which can be even chronic in a small set of cases. So unfortunately, also, all the current risk classifiers, even the most sophisticated, are prognostic but not predictive. But a couple of years ago, there were...
it's 6:30 they're doing, but I'm not sure
I'm almost finished. You can see...
I'm not sure I want to, given the weather.
Yeah. Okay.
No, I won't. No.
How much I have on more?
Silvia, go ahead. Just continue.
Okay, as you can see here, if you look at ctDNA, you can see that it's not only in the after surgery, but even after adjuvant therapy, and especially if you do serial liquid biopsy, it really differentiates positive versus negative. And so this is how we designed the Pegasus. We use a control arm, and I will not explain you why, but it's an interesting reason. And then those that were positive, we treat with capox, negative, we treat with cape, and answer the question, can we use ctDNA to downsize treatment? And then we can also use, we also asking, can we leverage ctDNA as a marker of chemoresistance? Because after 3 months of capox, if you were positive and you're still positive, what are the chances that you will become negative? 0. So we are switched. This is really a truly metastatic patient, even if micrometastatic. So we're treating with the second line standard of care for metastatic treatment, which is FOLFIRI. Okay. And we're also doing serial ctDNA tests to make sure to catch the false negative. We started the trial in mid-COVID, so at the beginning of the COVID. We started doing the regulatory in February, I mean, last year. Can you believe it? I mean, I really picked the right year. Anyhow, we have now opened the trial and accrued 25% of the patient, and the situation is that it is here. Pegasus and Alpha Omega are also, however, important to tackle the complexity. Again, this is something that I always thought was kind of crazy, that we think that we, with a monotherapy or even a polychemotherapy, try to solve the complexity of cancer. And I've been thinking this since the Bob Weinberg paper. And in addition to that, we are also not prepared conceptually, because everybody is working in a kind of a silos, you know, and they're not talking to each other. So what we designed, the idea was that Alpha Omega plus Pegasus could help tackle this complexity. How? Because from the very beginning, and at the moment in which we are collecting also the live tumor, it differentiates patient into born to be bad tumors to born to be good tumors, so tumors that are potentially cured. So these are phenotypically identical patients, so they must be biologically different. So we are able to study them prospectively and tackling them with a variety of information. So we are getting a kind of a holistic attitude, which is not going around, you know, doing OMS, but just doing different and completely, and looking at the different aspect of the tumor in the same tumor, in the same patient, under the same umbrella of trial. And the same thing we're doing at introducing a third level, which is the reason why we did the retrospective Alpha Omega. So looking at the possibility of using algorithm of artificial intelligence to mine the combined wealth of information that is collected by the radiology imaging and by the digital pathology imaging. The radiology imaging we harvest and we use now with the RECIST only 5% of the information that is captured by CT scan or MRI. And if we could couple that with clinical markers and molecular markers, we can come up with really integrative biomarker and not just a prognostic biomarker. And we have already, you know, obviously these kind of things you cannot do without a lot of money. And we are running for the last horizon and the new cancer mission. But we are started already the feasibility run and obviously in order to do this, we had to put up a super data lake, because if you produce a lot of data, then you have to have a way of analyzing. And first, even then analyzing in an integrative way, you have to store it. And so my conclusion are here, which I'm a Disney girl, and my favorite Disney movie is Cinderella, and my favorite song, it's the one that says, we can do it, we can do it. And that's what I think we can. I think we can. I think that this is the future. I think it takes, in this moment, a lot of research participation, but that there are glimpses that I hope you have seen in what I've said that show you that it also will be important for the patient care. And with this, I thank my group, which is really working very hard in this circumstances of the pandemic. And this is our core CRC team. Last time we met Devisu all together at my house almost a year ago. Thank you.
Bravo. Thank you so much, Silvia. Fantastic talk. Really, you gave us tour de force educational 30 minutes on so many subjects. So I would ask you to turn your cameras on if you can. And please go ahead with the question.
Okay.
John, would you like to start?
That's what I sound on. Well, it's not really my area of expertise, and you know, most people know what I think, and have put it on with Ian and Lidie and Lisa on personalized, or whatever you want to call it, therapy. I am attracted by desensitizing treatment.
Mm-hmm.
And I wonder, you know, if you can educate me about the number of trials that you think really have given sufficient data to say that we shouldn't treat this patient. Because there's
they're all ongoing. This is, I mean, my Pegasus, I think it's the first or the second in Europe that tackles this question. And as you see, as you've seen, we only de-escalated treated, not avoided, because we're not sure that we can avoid but we know for sure that 50% of those patients that are in there, actually more, there are 70% of the patients that now, because of the screening, are that really do not need it. And we don't know who they are, but we know they are in there. You see the conundrum. And therefore, the only way is to do trials. My group mission in life, it's not to do large-scale trials. We are research people, and we are doing proof of concept trials. So Pegasus is being designed as a proof of concept trial, and that's why I used historical control. And I was able to do that just because because I'm using the Tosca trial, which is a humongous phase 3 trials that randomized 32 to 100 of patients between the pivotal question 6 versus 3 months of treatment with Capox or Folfox. Okay, so I have very, very solid data of how these patients are, and I can do a match 3 to 1. So that's Pegasus. Then there are a large trial in the Jane Tai in Australia. It's conducting and has already closed the trial in stage 2, in which they're asking, and we don't know the result. And these are classical trials, you know, conventional classical design. And she's running the phase 3 trial, in which she is de-escalating or escalating according to the liquid biopsy. And then all the major cooperative groups in Europe and in the States are discussing and are in the middle of doing large-scale trials of this type. Okay, so the answer is going to be here quite soon, and not too much soon, because as I'm saying here, the majority of patients are overtreated, and this has been so clear in the past. You know, in the question 6 versus 3 months, 12,000 patients have been treated in the last decade, not to answer the question, because there was no biology behind the 6 versus 3. That's my point. Precision oncology might not be the answer, but the concept in terms of targeted therapy, as we have seen it some. But the concept of precision oncology is that you have to have biology guiding the large trial is something that I'm really an advocate of this, and I would be love to be challenged by you or by Ian or by whoever. But I think that this is what the era that we have now. It's to merge biology with good clinical trial methodology. I mean, I invented with Alessandro large-scale clinical trial and the Cochrane in Italy. So you cannot accuse me not to be a randomizing person. I randomize the coffee that I take because I don't trust myself. But it's not enough to randomize. I mean, my philosophy, my thoughts now is that we are using evidence-based medicine to do bad science, to ask wrong question. Biology is offering us the possibility to ask good question and do it with a good clinical methodology. Sorry, I'm being provocative.
Excellent. That's what we want. So thank you so much, Silvia. Lydia, you're next.
Yes. First of all, I've learned, if I'm correct, that just, let's say, diagnostics by ctDNA is defined also as precision oncology. It's not only that's how you see it, or does everybody has that same idea of definition? That's fine with me, but for me, precision oncology was mainly about treating based on mutations, but that's the broader definition then? Do you agree on that, John?
Sorry. No.
Yeah, so do we all agree that like diagnosis by ctDNA is also part of the definition of precision oncology? Because mostly it's more narrow. It's precision oncology is just about treatment, treating mutations. Let's say it.
Well, taking the example that Silvia showed, and I didn't hear your question because I was reading Elizabeth's comment on the discussion that this has been done in to de-escalate in breast cancer. I mean, I think that this is an important aspect of improving the quality of care.
Yeah.
You know, maybe the drugs don't improve the quality of care, but not having the drugs certainly might do so.
Yeah.
So these are good. I mean, I think in the arguments against so-called precision or personalized medicine, the arguments are that there isn't enough data being generated to look at options like this. And I like very much the idea that you're doing it.
Mm-hmm. By the way, I totally agree with the definition, the NCI definition of precision oncology includes diagnostic. So I think that precision is anything that will allow you to differentiate between what you should do and what you shouldn't. I mean, precision, it's that. I mean, it's a semantic discussion. And by the way, I totally agree with Elizabeth that in breast cancer, things are much earlier because of the oncotype. I mean, because of the transcriptomic and the RNA classification. They are less precise, perhaps, because but they have allowed to do this. And I think that the de-escalation, it's really going to be a major focus of the next trials. It's going to take several years, but we're going to go there.
And then I like very much the de-escalation trials. And there, my question is, how do you get them funded? Is that with public money, I assume, or how does all the trials you mentioned and yours in particular?
Okay, I have we have been funded by this charity a lot, but I'm also very good at negotiating. I come from a family that they are good negotiators, so I must have it in the gene because, for instance, in Pegasus, Guardant Health is paying for all the ctDNA test, so it's, you know, more or less 2 million euros of tests. And also in my next trial, which I've already designed, in which I'm coupling the diagnostic with the target therapy, which is called Pegasus Precision Oncology. I've done this the same. We actually got Natera, which is the other big provider of liquid biopsy test, to pay for the test. And the drugs we get from the pharma, free of charge. So it takes a lot to do this kind. I work 12 hours a day convincing people, but so far we succeed, and let's hope and keep our finger crossed that we can do it in the future as well.
Okay, and hopefully indeed within the cancer mission, this is the type of trials that I think are worth having support from the cancer mission. We certainly would be willing to.
Well, telling you the truth, last year I did the Horizon with the next generation Pegasus, and we got through the first step, and then we lost for 0.5% the second. So I don't know, but it was not a good call. So. And
then my last question, sorry.
There are other people, Lydia. I perhaps.
Yeah. Okay.
You can come back later.
Yeah. Okay.
So, Michael.
Yeah. Hi, Mike Kolodziej. I appreciate your talk. I am familiar both with the challenges in deciding which patients with stage 2 colon cancer to treat, and I'm also familiar with some of the preliminary evidence regarding circulating tumor DNA. So here's the question I have. You know, as I think about this from the perspective of both a treating physician as well as somebody who worked at an insurance company trying to decide whether or not this technology ought to be used routinely, I reflect on how we report clinical trials, success or failure, based on performance within a population, right? So we're looking at how good the test performs within the population, as opposed to what a provider needs and what a health plan needs, which is the performance in an individual, right? So let me see if I can articulate this.
Mm-hmm.
You know, we've got a ton of evidence now that on average, PD-L1 expression is a pretty good predictor of response to therapy with IO.
Mm-hmm.
However, there is almost nobody who's using that to make a decision in an individual patient. Now, you may think that's wrong. The studies are very convincing that if you have expression over a certain level, and the reason is we're not comfortable saying that this is good for 90% of patients. It has to be good for almost every single patient. How do we get beyond that? I mean, how do we get comfortable with the idea that this, let's say circulating tumor DNA does in fact in a population identify a cohort of patients likely to either benefit or not benefit or require a change in all that stuff. Let's say it all turns out to be right. How do I translate that into making a decision for an individual patient?
I don't know. That's an interesting question, because it's a perspective that in Europe we don't have, because we do not have the problem of the provider. So I personally think at myself, if I had to come up with with a colon cancer, and I would do a ctDNA, and if the ctDNA was negative, I would not go choose treatment. So I think it's simple as that. And, you know, even if it's immunotherapy, I mean, why we're using the cutoff at 1% rather than 20, when we know very well that 1% is really nothing, that it's an average when you see it, okay? That's because the industry, if you get the 1% cutoff, treats with a very expensive drugs a lot of people. Whereas if you take the 20% cutoff, you know, and even going higher, but there is a cursor, obviously, because there is a situation in which you're going to have a lot of false negative or false positive in that case. But it's driven by the industry. It's driven by in this case, the provider of the drug that has to make a lot of money out of it. I wouldn't go through immunotherapy if I had just 1% or 1.2%. I probably certainly would do it if I would 50, and if I was 20, I would not know what is at 10, but at a certain point you have to do rock curves and go with that and choose a cutoff. We've done that all the time.
So the perspective is, I think introducing finances into this is irrelevant. Let's take the example of hormonal therapy in breast cancer, which costs next to nothing.
Yeah.
We give hormonal therapy to early stage estrogen receptor positive breast cancer for a minuscule benefit. Minuscule, right? Doesn't cost any money. Minuscule. Why do we do that? Because we want to extract benefit for every patient who might benefit. And I would argue, for example, that global immunization for COVID is really only going to protect a small percent of the population from death. And yet, we're immunizing everybody, which, thank God I'm immunized. I'm so happy I can't express it to you. But I'm just saying to you that the philosophy is we do not want to deny benefit from any patient who might benefit. And that's why these kind of studies are problematic, because the answer is unlikely to be binary. It's just not going to be binary. And I think as a consequence, we're going to have to make a decision. We got to make a decision.
I think that what you are saying is that it's something that I've been thinking for a long time, which is that we are assessing the risk of toxicity, which is always associated with a drug, with the risk of death, with two different measures, which is okay if you do it because the decision is made by medical oncologists that think in terms of metastatic disease first. So in the metastatic disease, the probability, even a small probability of response, and even, you know, the hope of cure, which happens once in a while. You know, I said I have one patient out of 27 that's still alive and kicking 7 years after starting the therapy with Heracles. Okay, that has a tremendous importance in my eyes and in the eyes of the patient compared to the toxicity I'm offering, okay? Whereas when you work in the adjuvant disease, you have to think with a different mentality, but because there, the advantage are very small, but the toxicity is very real. And colon is a very good example for this, because the toxicity of oxaliplatinum is really life threatening, no, not threatening, life, quality of life threatening. You know, patient you know, they cannot hold a cup because of the neurotoxicity.
Yeah, no doubt.
And however, as I said, we spent 12,000 patient worth for deciding 4 months versus 5 months, which is crazy. And the difference is at 5 years is 0.4% in survival, 0.4% absolute, which is nothing.
No,
so
listen,
I think that we need to switch our things, and we need to. That's why there are prompts, and you know, we should ask patient, and not make the decision for them. But Elizabeth talked about this. Sorry, you know about more than I do about this.
No,
sorry,
so the interesting question,
thing.
So to me, the interesting question is this. You know, the circulating tumor DNA right now is probably the first iteration of what will become a much more sophisticated biomarker identification for prognostication, right? So it's not just going to be whether or not there's circulating tumor DNA. It's going to be what that circulating tumor DNA contains, right? Certain profiles are likely to going to be different risk than other profiles. Some of them are going to have P53 mutations. Those people are going to do very poorly, and they'll be chemotherapy resistant. I mean, it's just, my fear is this. We're falling into the trap of doing very large randomized clinical trials that take forever to complete. What we need to do is we need to do 50 patients, look at the signature, change the trial, do it again, and do it fast. Rapid iteration. Right?
That's exactly what I'm trying to do. Yeah, I agree with you. But this has to, the large trials have to be done, but they have to be, I don't know how you do this. Large trials that, adaptive trials have this idea that you start low and then you change. But the question is the biology behind, when are you, the things that I think now it's difficult for me to do is, when do I go with a large trial? What is enough knowledge? What is enough understanding of the biology behind to do really the effort of going into a large trial, which you would not end doing if you're really correct in what you're looking. You didn't need randomized trials for antibiotics at the beginning.
Thank you. Thank you. Very good challenge from Michael. So Ian, you next, and then we can take another question from Lydia.
Yeah, first of all, I agree with what Elizabeth has said in the panel, and she may want to speak to that. Secondly, Silvia, I'm all for biology trying to give us insight in what to do well. I think my and John's criticism of the current sort of rather narrow definition of personalized medicine or precision medicine, where patients are being persuaded that having their DNA sequence, and of course that's a sequence of one part of a tumor at one time, is not productive, and that's what's mainly being done.
Mm-hmm.
I particularly like, these graphs you showed of survival based on positive versus negative ctDNA after surgery. I think that's very meaningful. But again, and Elizabeth has said this in the chat. You said if you didn't have a positive ctDNA, you wouldn't undergo adjuvant therapy. But there is, I can't remember the actual numbers, but say there's perhaps a 10 to 20% relapse and ultimate death from disease, even though it doesn't have ctDNA early. And that seems to me to be a pointer for doing a different type of trial. Maybe you wouldn't take oxaliplatin with its toxicity, but maybe you would go back to a much simpler regimen, because clearly there are a proportion of those patients who have the risk of recurrence. So I think it is how much information you need to prevent a treatment that might still save lives, and as you say, balanced against that toxicity. I like very much the idea that for those patients that you do have a high risk, that you are randomizing against a sort of biologically derived directed treatment that includes appropriate targeting, where you actually find a mutation used in the widest sense of that term, against standard therapy. I think that absolutely needs to be done, because to my mind, the current generation of basket trials, some of which you mentioned, haven't shown that we're doing better than if we took a drug off the shelf, for example. So I think it's good that you pointed that out. I don't want to be named as somebody who's against biological advances driving treatment. I absolutely believe they should. I just need to think that, as you said, we need to recognize that the cancer is a lot more complex than being a simple mutation in a driver gene from one biopsy that is a good guide to treatment. That's failed, so we need to move on.
Okay, you, as usually, has been very wise. First of all, false negative. The slide that I've showed you are with the old test. The new ones are much more sensitive, either because they have upgraded the techniques of doing NGS, or most like in Pegasus, because they've added a layer of epigenetic marker on top of the genomic one. So we are looking at two different things. In addition, we organized to do 3 tests, one after the other, spaced in a month. So the worst that can happen is two things can happen: that you pick up second tumors unrelated, and we've done that twice in the first 35 patients. So that's a phenomenon that it's much higher than what you would suspect. And the second thing is that even if you have a false negative at the first or second test, the probability that you take it up to the third, that with 3 tests you get it, it's up above 95%. So it's well above the gain that of doing chemotherapy to all. And you could switch immediately, you know, and you can delay that only a month and a half. So that's a reasonable risk. I think what I'm saying is that this is giving us the opportunity of thinking in terms of risks and not absolutes, like also Michael was saying before, which I think it's an important thing, you know, to start doing with the patient. And the other thing is that, absolutely, you're right. My next generation trial, which is already designed, and unfortunately I don't have that slide here, is asking the question. It's randomizing also the negative patient versus nothing, or the conventional treatment, physician conventional treatment. And in the treatment arm is asking the question of targeted therapy, because I am absolutely sure that you cannot cure a metastatic disease, which is heterogeneous and with an evolution and co-evolution with the immune system, which has gone berserk with, you know, HER2 alone, even if the patient is truly addicted to HER2. But perhaps if you are working early on in this disease, there are some data that suggested that if you pick the disease when there is only a trunk mutation and the driver mutation, and the heterogeneity is still there, but not that pronounced, perhaps you can really do something. So we're bringing targeted therapy frontline in adjuvant therapy. That's going to be a year from now, if everything goes well, which... But it goes into your direction. I truly think that we have to be innovative if we want. I mean, in colon cancer, we've been doing the same things for 20 years. Before dying, I want to see something new, even if to say it's not working.
Thank you, Silvia. Liddy, the floor is yours.
Maybe Lisbeth first.
Elizabeth just keeps writing things, but I cannot. I'm sorry to leave. No, this is somebody.
You have to leave, Elizabeth. I will too.
I wrote some comments in the chat. I thought 2 big messages that I got from your talk. Amazing that you've been able to kind of pull all this together in the sort of the time of COVID. Research in the time of COVID will be a novel. I think one of the observations I would make about how oncology has evolved over the 35 plus years I've been involved is that we've gone from the days when if you only had one tool in your toolbox, you chose very well on whom and when to offer that to patients. Now it seems oncologists have an endless stream of ideas of stuff to do, and they're looking at ways to justify pulling things out of their toolbox to do more and more and more. And I really think it's important to take a step back and say, when should we stop doing, when is treatment really futile and we have to have hard conversations with patients? And who do we not need to treat because they're doing so well? And those two groups, those in whom treatment's futile and those for whom prognosis is so good that treatment's not needed, I think are the 2 big gifts that might come from precision oncology. So instead of thinking about it as what more we can do if we test and test and test and pull drugs out of the air, think about it and who can we spare treatment because they're either going to do too well or because in whom it's going to add toxicity with no real hope of benefit. So I would add that. I would also say the big challenge in backing off treatment lies with the fact that we're so subspecialized that for a breast oncologist, giving up 1 or 2% in long-term disease-free survival is horrifying. And that is a difference in outcome that for, let's say, a GI oncologist or somebody doing prostate cancer, they would never have adopted that treatment to begin with on an absolute basis.
Mm-hmm.
And so the measures of getting back to some of the comments about what degree of difference in outcome is worth not doing something or doing something, there's going to be different perspectives based on the subspecialty area and the traditions that have been adopted over time, which makes it extremely hard to get rid of treatments in, let's say, breast cancer or Hodgkin lymphoma when outcomes are very good. And the gains that have led to adoption for everyone of new treatments have been fairly tiny on an absolute basis, versus diseases like, let's say, pancreas or some GI cancers where the outcomes are relatively poor and you're looking for an absolute gain that's greater.
Mm-hmm.
By the same token, the only other thing I wanted to comment on was ctDNA, sort of the technology that does everything. It's kind of like the new ovens that you can buy that can cook all sorts of things and clean cells. The technology that does everything, it can offer some prognostic information. It can be a cheap biopsy. I can remember the days, I date myself, when my nephrologist teachers used to tell me that the urinalysis was the poor man's renal biopsy. And I think ctDNA analysis are going to become the poor man's tumor biopsy. But they have their limitations as well. But they can tell you if there's circulating tumor DNA and what that might mean in terms of prognosis, good or bad. It can be a way of following disease. And I think that's where it's most dangerous, because of course, as you follow disease and you say, ooh, ctDNA is rising, we better start a treatment even before people have anything there. That just adds toxicity without any proven benefit. But by the same token, you're showing in stage 3 cancers, there's patients without ctDNA. Maybe they don't need colorectal cancer adjuvant treatment. Maybe in stage 2, there are patients with it who do need it. And so maybe it's going to become a new staging tool, but that's something that will require lots of data and lots of information, maybe even from ongoing randomized trials where people were clever enough to collect it so that we can evaluate it. Anyway, I'll shut up now and put most of my comments in the chat. And it's nice to see you.
Yeah, well, the last, Jane Tye, as I said, Jane Tye has just closed the trial. It's going to take another 3 or 4 years to evaluate, but they've just closed the trial in the stage 2 with the idea of treating those, where they were treating those that were ctDNA positive. So I think that's a humongous trials because, but 20% of those patients do relapse, and so they should be treated. And the data on the untreated patient are even more. They have something like a 17 hazard ratio, those that are positive. Practically all of them are in relapse after 2 years. So I agree with you. I agree. This is going to be a little bit of a panacea, and it's going to be very, very, and I'm not touching with a 10-foot pole the idea of anticipating treatment on that. I'm at the stage I want to avoid treatment when it's not needed. And I totally agree. And the reason why I did Pegasus and Alpha Omega was to create that extreme, that you were saying, the exceptional responders and the exceptional non-responders. Everything in between, it's a gray area. But these two, if we are so different biologically, that if we study and exacerbate this difference, we may find something that it's more interesting than just looking at everybody. And that's what Pegasus coupled with Alpha Omega is allowing us to do. To look at the extreme, which phenotypically are identical. Even genotypically, these patients are the same. There are KRAS patients that are doing very badly and KRAS patients that are doing perfectly well. And you don't know why. So we have to look at other things. We have to look at the relationship with the microambience of the tumor and of the host, so the microbiome and the immunostate income. If we do not understand complexity, we will not ever, ever cure cancer. It's probably impossible, but at least mitigate its toxicity.
Thank you, Silvia. So Lydia, last question from you before we wrap up.
Very short question regarding the Heracles trial. Do you have overall survival data on that?
Yes, I don't remember. That's an old trial.
Okay.
I can tell you that it was, no, I can't remember what it was. We do have it. It's been published also.
Okay.
Recently in Annals of Oncology. Sorry. We did also other trials. We did the trials with pembrolizumab and trastuzumab that had the same results, PFS-wise and survival-wise than the first one, but response rate was not as good. And we still don't know why. Other people have found that that combination is as good as ours. And there's many trials ongoing now. It's becoming mainstream, and fortunately, it's gonna eventually do a randomized trial.
Thank you.
So Silvia, thank you so much. Absolutely amazing work you're doing, and what very much we look at Consilium is there's a huge lack of activity in post-registrational phase. So we're really lacking confirmatory trials that you're doing, further investigations to really improve care for patients, and how these trials are designed, how quality and rigor of those trials. We have amazing examples and we have very disappointing examples. So we really need to try to push and see what we can do with the arsenal of treatments we have right now to manage our patients better and not just rush for new cures, moonshot type of solutions. And we're massively lacking funding, we're lacking ambition in terms of, because discoveries in that space are limited, complicated. So thank you so much for doing this work, for educating us, for helping patients, and for teaching us about trial design. So I just want to say a few words in terms of what's coming up. We're very much on oncology theme, thanks to John. So in 2 weeks, we have a talk about European Horizon policy in cancer. And then we have Caroline Dive speaking about liquid biopsies in lung cancer. So many of you may be interested in this topic. And then we have Francois, who will join us in July to talk about trial designs. So we worked with Francois a lot on this area. Very much looking forward to learning from all our future speakers. And thanks so much for joining us again.